Hello everyone, I'm Stefano Pagliara. I'm a senior lecturer in Biosciences. I work at the Living System Institute at the University of Exeter. The subject of my research is studying antimicrobial resistance, looking at one cell at a time. And the way we do this is by using some novel cutting-edge technologies that we have developed. We have recently discovered that there are no two identical cells on Earth. So all cells are slightly different from each other, and this has important consequences for antimicrobial resistance, but also for cancer development and for the development of embryonic stem cells. So cells can be different in the way they grow. Some cells grow very fast and some others grow more slowly, but they can also be different in the way they respond to stress, like heat stress or chemical treatment. So the specific topic of my research is on bacteria and bacterial infections. There are trillions of bacteria around us, and there are actually more bacteria within our body than our own body cells. Most of the bacteria that live in us or on us are actually beneficial for us. They perform very important metabolic functions, for example, by breaking down complex metabolites that our body cannot assimilate. But from time to time, bacteria can become bad for us by causing infections. For example, when we have a cut in our skin, bacteria can penetrate and cause an infection or cause, for example, lining infection or a sore throat. And so in those cases, sometimes our immune system is good enough to kill the bacteria who are causing the infections, but sometimes we need some chemical treatments, and we do this by using antibiotics, which have revolutionized medicine in the last 50 years because are very good in cleaning bacterial infections. However, bacteria have become very clever because they have been exposed to antibiotics for a long time, and so they have found several different ways for surviving antibiotic treatment, and that poses a daunting threat for us because that means that the weapons that we have available for killing bacteria are actually now less and less potent. And so what happens is that, for example, we go to a GP and they prescribe an antibiotic for us, but that doesn't work. And so we need to go back after a week to get a second antibiotic, and it's a bit of a waste of time for the GP, but also quite painful for the patient. And so the way we approach this problem is by looking at bacterial infections and bacterial treatment with antibiotics one cell at a time. And the way we do this is by using microfluidic devices like these ones. So the one on my left is a very simple microfluidic device made of a layer of plastic. And in there we have written 3 letters, LSI, which is the acronym for Living System Institute, which is the place where I work. And you see the nice red in there is given by a colorful dye that we have used for visualization only. This instead is the microfluidic device that we actually use for our research. So it's made of a piece of glass. On top of it, there is a piece of plastic, which is called polydimetic siloxane. And the nice thing is that we can write in this piece of plastic, and we can create some microchambers in which we trap the bacteria. And then in these chambers, we can keep the bacteria over time and investigate their response to antibiotics. The way we do this is by placing this device on a microscope so that we can image each single bacteria over time. And then we can inject whenever we want into the device different antibiotics, so 4 different antibiotics to be tested against this bacteria. And from this other tubing, we can collect the whatever is secreted by the bacteria and all the dead bacteria. So with this device, we can look at bacteria over time. We can look at their response to the antibiotics, and we can very quickly tell whether an antibiotic is actually effective in clearing a bacterial infection or is not good and shouldn't be prescribed. So by using these kind of devices, we have found that besides developing genetic tools for surviving antibiotics, bacteria can actually phenotypically survive to antibiotics. This means that even though there are no changes in their genome, they have found a clever way to survive the antibiotic. For example, by changing the properties of their membrane, they can exclude antibiotics entering into the bacterium. or by using some specialized proteins that pump out the antibiotics, they can decrease the amount of drug within the cell and in this way survive the antibiotic. So we have found that some specialized bacteria that we have dubbed sleeper cells can survive antibiotic treatment by simply going in a dormant state, and these pose a serious threat for our health. For example, when seafood is farmed, it's a low temperature, and some bacteria go in a dormant state. But then what happens is that when we eat the seafood, the bacteria found a nice cozy environment at 37 degrees with a lot of nutrients around them, and in that way they can wake up from this dormant state, from this sleeping state, and they can infect our gut. Another strategy that we have found the bacteria use for surviving antibiotics is that they can play with the amount of antibiotics that enter and exit the cell to lower down the amount of antibiotic within them. And this can change from two daughter cells to cells that are one next to each other. And so what we have done is that we have devised a platform that can quickly tell us whether an antibiotic is entering into an individual cell or not. And by testing several antibiotics in parallel, we can inform of which antibiotics should a GP prescribe because we think that antibiotic is going to work, and which antibiotics shouldn't be prescribed at all because they cannot even enter into the cell, so they cannot find a bacterial target to kill the cell. So with our research, we are now in a position in which we can both inform the development of novel antimicrobials to clear bacterial infections, but we can also very quickly inform clinicians and GPs on which antibiotics should be used for that specific patient, for the bacteria that are taken from that specific patient.